Hello everyone, today I will be discussing about basics of transcription and translation. So, what is transcription? Transcription is a synthesis of one RNA molecule using one of the two DNA strand as a template by enzyme RNA polymerase, that you can see clear in this picture. What is transcription? So, transcription is a synthesis of one RNA molecule using one of the two DNA strand as a template by the enzyme RNA polymerase. You can see in this flow diagram very clear the central dogma, the DNA codes for RNA and RNA codes for protein. So, first we will take prokaryotic transcription. So, first we will take prokaryotic transcription, the RNA polymerase, the origin and prokaryotic promoters, the initiation, elongation and termination. Then prokaryotic termination signals, prokaryotic transcription product. RNA polymerase is the enzyme responsible for the RNA synthesis is DNA dependent RNA polymerase. The prokaryotic RNA polymerase is a multiple subunit of protein, that is 480 kD. That requires no primer for polymerization, that requires DNA for activity and is most active with a double stranded DNA as a template, and 5' 3' synthesis that require magnesium for RNA synthesis activity, that lacks 3' 5' endonuclease activity, and the error rate of nucleotides incorporation is 10 to 4 to 10 to 5, usually are multi subunit enzyme. Here in this picture you can see the core RNA polymerase with sigma factor, that is RNA polymerase holoenzyme. If we see the RNA polymerase of E. coli, you can see here the 5 different subunits, that is alpha, beta, beta dash, theta and omega. In this pictorial representation, you can see here very clearly the process. So, what is eukaryotic promoter sequences? The promoter, enhancer and activator. If we see the structure of bacterial prokaryotic promoter region, the TATA box or now box, this is a stretch of 6 nucleotides centered about 8 to 10 nucleotides to the left of the transcription start site. 35 sequence are second consequences nucleotide sequence, that is 5' TTGA CA to 5' 3' is centered about 35 bases to the left of the transcription start site. 5 steps are there. First is promoter binding, then DNA unwinding, then RNA chain initiation, then RNA elongation and RNA chain termination. We will take one by one. In initiation, RNA polymerase recognize the TTGACA region and the TT region sequence, then open the DNA duplex. The unwound region is about 17 BP. You can see here the double helical DNA, then unwound DNA, that is 17 BP opened in RNA polymerase. The first nucleotide on RNA transcript is always purine triphosphate. GTP is more often than ATP. The PP, GP and OH structure remains on the RNA transcript until the RNA synthesis is completed. The three molecules from a transcription initiation complex, no primer is needed for RNA synthesis. The sigma subunit falls off from the RNA polymerase once the first 3' 5' phosphodiester bond is formed. The core enzyme moves along the DNA template to enter the elongation phase. In elongation phase, the release of the sigma subunit causes the conformational change of the core enzyme. The core enzyme slides on the DNA template toward the 3' end. Free NTP share added subsequently to the 3 OH of the nascent RNA strand. RNA polymerase DNA segment of 40 and nascent RNA form a complex called the transcription bubble. The 3' segment of the nascent RNA hybridizes with the DNA template and its 5' end extend out the transcription bubble as the synthesis is processing. You can see the transcription bubble here in this pictorial representation. Nascent RNA is very clear. Here also it is very clear, the processes you can see in this picture. The RNA polymerase, the removal of sigma subunit. In termination, the RNA polymerase stops moving on the DNA templates. The RNA transcript falls off from the transcription complex. The termination occurs in either P dependent or P independent manner. Termination function of P factor, P dependent termination. You can see here the P factor hexamer is a ATPase and helicase. The P independent termination. In P independent termination, the termination signal is a stretch of 30 to 40 nucleotides on the RNA transcript, consists of many GC U followed by a series of U. The sequence specificity of this nascent RNA transcript will form particular stem loop structure of terminate the transcription. In P independent termination, this picture is very clear. Then inhibitors of transcription, it binds with the beta subunit of prokaryotic RNA polymerase and thus inhibit the elongation phase of transcription. The tricycling system of actinomycin di-intercatalysis between adjacent GC base pair and the cyclic polypeptide arms fill the nearby narrow groove and inhibits elongation phase of transcription. Actinomycin D. So, this is the eukaryotic transcription. Eukaryotic RNA polymerase 1 transcribes synthesizer 28S RNA, rRNA, 18S rRNA, 5S rRNA, RNA polymerase 2 transcribes messenger RNA, some SNA RNA, RNA and some SN RNA. Polymerase 3 transcribes tRNA, 5S rRNA, some snRNA. In RNA polymerase 2, RNA polymerase 2 is the central to eukaryotic gene expression and has been studied extensively. RNA polymerase 2 is a multi-subunit enzyme with 12 subunits. RNA polymerase 2 requires an array of other proteins called transcription factor, that is TF factor 2, in order to form the active transcription complex. Eukaryotic RNA polymerases, you can see here the all three subunits. Eukaryotic transcription promoters. In eukaryotic transcription promoters, much more complex than those found in a bacteria. These are sequences located at the upstream regions of coding strand. Mutation of this region usually significantly lower the rate of transcription. First is TATA box, that is Hogness box, very similar to the prokaryotic TATA box, except the sequence is slightly different, that is TA, T, AAA and is located between 25 to 30. Second one is CAAT box, located between 70 to 80, always contain CCAAT. GC box is usually has the sequence GGGC, GG and is typically found at 110. Enhancer. Now, the enhancer elements are the sequences located in a variety of regions of a gene, both upstream and downstream of the transcription start site and even within the transcribed portion of the some genes. Enhancer increases the transcription rate by several fold. You can see here in upstream also and in downstream also. transcription factors are RNA polymerase II does not bind to the promoter sequences directly. RNA polymerase II associated with 6 transcription factor TF2A, TF2B, TF2D, TF2E, TF2F and TF2H. Now, pre-initiation complex, that is PIC, TBP of TF2D binds TA box, that is in 10 sequence, and TF2A and TF2B bind TF2D, TF2F, RNA polymerase complex binds TF2B, and TF2F and TF2E open the dsDNA, that is helicase and ATPase, and TF2H completion of PIC, that is pre-initiation complex. RNA polymerase II to DNA. Phosphorylation of RNA polymerase II, TF2H is protein kinase activity to phosphorylate CTD of RNA polymerase II. There the CTD is the C-terminal domain of RNA polymerase II. Only the RNA polymerase can move towards the downstream, starting the elongation phase. Most of the transcription factors fall off from PIC during the elongation phase. Here you can see clearly the RNA polymerase II. Then next is the termination. When the RNA polymerase transcribes the terminator region of the DNA, the polymerase releases the mRNA. The termination sequence is AA, TAAA followed by GT repeats. TF2F remains associated with RNA polymerase II throughout elongation. The activity of the RNA polymerase II is greatly enhanced by protein called elongation factors. Polymerized mRNA polymerase. Then translation. Translation is the assembly of polypeptides on a ribosome. Living cells devote more energy to the synthesis of protein than to any other except of metabolism. So, what is translation? Translation is the assembly of polypeptides on a ribosome. Living cells devote more energy to the synthesis of protein than to any other aspect of metabolism. Then what are the components of translation? mRNA. mRNA in eukaryotes made in the nucleus, transported to the cytoplasm, and in prokaryotes transcription and translation occur concurrently, and in tRNA next is the tRNA. Adapter molecules that mediate the transfer of information from nucleic acid to protein. The next component is the ribosome. Ribosome manufacturing unit of the cell located in the cytoplasm contain ribosomal RNA and protein. Next component is the enzyme that required for the attachment of amino acid to the correct tRNA molecule and for peptide bond formation between amino acid. protein soluble factor necessary for proper initiation, elongation and termination of the translation. Then mechanism of translation. The 3 steps of translation are: first is elongation, initiation sets the stage for polypeptide synthesis, then elongation causes the sequential addition of amino acid to the polypeptide chain in a collinear fashion as determined by the sequence of mRNA. And the third step is termination, that brings the polypeptide synthesis to a halt. So, initiation codon is an AUG, that is towards the 5 prime end of the messenger RNA molecule that is being translated, not the first 3 nucleotide. It determines the reading frame. In prokaryotes, there is a conserved region about 7 nucleotide upstream from the initiating AUG. This region contains a 6 nucleotide sequence signed Dalgano box, that is AGG AGG. The Shine Dalgano sequence is complementary to a region at the 3 prime end of the 16 rRNA of the small subunit. Base pairing between these complementary sequences stabilizes the binding of a small ribosomal subunit to the messenger RNA for proper assembly. Signed Dalgano sequence, that is starting codon, you can see here the 16S RNA and messenger RNA, and the whole process is depicted in this pictorial representation. Here also you can see in eukaryotes. Then next step is the elongation. At the start of elongation, the messenger RNA is bound to the complete 2 subunit ribosome with the initiating tRNA in the P site and the A site free for binding to the next tRNA. The ribosome moves along the mRNA in a 5 prime to 3 prime direction in a stepwise processes, recognizing each subsequent codon, and the peptide 1 transferase enzyme then catalyzes the formation of a peptide bond between the free and terminal of the amino acid and the A site, and the carboxyl end of the amino acid at the P site, which is the actually connected to the tRNA. This disconnects the tRNA T-met from the amino acid, and the tRNA at the A site now carries 2 amino acid with a free N terminal and the carboxyl terminal of the second AA connected to its tRNA. This is the translation elongation. Chain elongation. Chain elongation translocation. During translocation, the peptide tRNA remains attached to its codon but is transferred from the ribosomal A site to the P site by the unknown mechanism. The vacant A site now contains a new codon and an amino acyl tRNA with the correct anticodon can enter and bind. This process repeats until a stop codon is reached. Elongation and translocation are the similar in eukaryotes except for the differences in number and type of elongation factor and the exact sequence of event. In both prokaryotes and eukaryotes, simultaneous translation occurs. New ribosomes may initiate as soon as the previous ribosome has moved away from the initiation site, creating a polyribosome या polysome. An average mRNA might have 8 to 10 ribosomes attached at a given moment. You can see here the ribosome movement here. Then termination. When the ribosome encounters a stop codon, there is no tRNA available to bind to the A site of the ribosome. Instead, a release factor binds to it. The details are not very clear, but once the release factor binds, the ribosome unit falls apart, releasing the large and small subunits. The tRNA carrying the polypeptide is also released, freeing a polypeptide product. This is the pictorial representation, a step by step. The future prospects. Future prospect is expanded role of RNA. Originally, the central dogma emphasized a unidirectional flow of genetic information from DNA to RNA to protein. However, research has revealed the importance of RNA in additional roles beyond mRNA. Other prospects in epigenetics. Epigenetics involves heritable changes in gene expression that do not involve alteration to the underlying DNA sequence. In CRISPR-Cas9 and gene editing, the development of CRISPR-Cas9 and another gene editing technologies has allowed scientists to manipulate DNA sequence directly. This has significant implications for modifying gene to treat genetic disease, enhance agricultural crops and even potentially create synthetic life forms. It is used in synthetic biology also. Synthetic biology aims to design and construct novel biological system and organism for specific purposes. It is used in biotechnology and medicine also. Advances in our understanding of the central dogma have had profound implications for biotechnology and medicines. This includes the development of biopharmaceuticals, gene therapies and personalized medicine approaches. Origin of life and astrobiology. The central dogma is also relevant to the study of origin of life and astrobiology. Understanding how genetic information can be transferred and replicated in different environments is crucial for assessing the potential for life beyond Earth. It is used in quantitative biology and system biology also. The integration of mathematical modeling and computational approaches with molecular biology is allowing scientists to analyze complex biological system in a more holistic manner. Future prospects of transcription and translation, I am repeating again. It has a expanded role of RNA. Originally, the central dogma emphasized a unidirectional flow of genetic information from DNA to RNA to protein. However, research has revealed the importance of RNA in additional roles beyond mRNA. It can be used in epigenetics also. Epigenetics involves heritable changes in gene expression that do not involve alteration to the underlying DNA sequence. It is used in CRISPR-Cas9 and gene editing. The development of CRISPR-Cas9 and other gene editing technologies has allowed scientists to manipulate DNA sequences directly. This has significant implications for modifying gene to treat genetic diseases, enhance agricultural crops and even potentially create synthetic life forms. Used in synthetic biology also. Synthetic biology aims to design and construct novel biological system and organism for specific purposes. Used in biotechnology and medicine, advances in our understanding of the central dogma have had profound implication for biotechnology and medicine. This includes the development of biopharmaceuticals, gene therapies and personalized medicine approaches. Origin of life and astrobiology. It is used in origin of life and astrobiology also. The central dogma is also relevant to the study of the origin of life and astrobiology. Understanding how genetic information can be transferred and replicated in different environments is crucial for assessing the potential of life beyond Earth. Future prospects in quantitative biology and system biology also. The integration of mathematical modeling and computational approaches with molecular biology is allowing scientists to analyze complex biological system in a more holistic manner.